The year 2020 was a game changer. The most imminent issue was probably the escalating pandemic of COVID-19 that changed the world and drastically altered our lives. As the iGEM team from the Technion, we don't just hope for better times, we choose to act. There are two common ways to get infected. The first is through inhalation of aerosols or droplets, and the second is through touching the facial area after contact with an infected surface. Our goal is to develop a product that will reduce infection caused by contact transmission. There are still many open questions about the SARS-CoV-2, and the comparison between it and the common flu is still frequent. For example, it has been shown that the coronavirus can survive on the skin for an average of 9 hours, compared to just 2 hours for the influenza viruses. Therefore, proper hand hygiene is important to prevent the spread of COVID-19 infections. The solutions that have been offered to us so far are gloves that are misused and thus can aid in viral transmission, hand sanitizer that only help at the moment they are used, and soap that is not as available as we wish in all times and places. In order to create a solution that bypasses those problems and that could actively help preventing the infection, we came with the idea to create a skin product containing particles that will display decoy proteins, akin to SARS-CoV-2. We figured out that our product needs to be user-friendly and accepted by society, with a short design cycle, safe to use, and the option to rapidly implement it when the next pandemics arise will be a huge plus. In search for social acceptance, we ran a survey, reaching over 1,000 people from 13 countries. We found that 68% of the potential consumers would like to use such a product, and the majority prefer the product to be in the form of cream. From a short design cycle, we found that using synthetic biology tools can be the perfect plan of action, due to the fact they are relatively fast to design and easy to modify on the move. Considering the safety of our final product was without doubt one of our design key aspects. That is why we decided to choose a non-invasive tool for skin application and make sure that unlike hand sanitizer, it will not damage the skin and its microbiota, a general name for the essential microorganism that lives on our skin. Lastly, for the possibility of future implementation, since our suggested solution is target-specific, the product had to be modular in a way that could make it useful for the next pandemics as well. SARS-CoV-2, like other coronaviruses, has a lipid bilayer membrane rich with glycoproteins known as spike proteins. This virus uses those spikes to infiltrate lung cells by creating a bond between them and a protein display on the human cell surfaces. This protein is known as ACE2. So what do we mean by decoy proteins? Essentially, they mimic the ACE2 receptors, but are engineered to be more attractive to the virus than the original ACE2. Once the virus is strongly bound to the decoy proteins on our skin, it will not enter our body when we touch our face.
In search of the best decoy candidate, we found two that fit best, each for its own reasons. The first choice was a mutated ACE2 protein. The mutations were designed in a way that would theoretically increase the affinity of the engineered decoy protein to the viral spikes. We have also been working on special antibodies. These synthetic single-domain antibodies are known as Sybodies and can be expressed in bacteria. They were designed and screened to find the variants most suitable to act as decoy proteins. In the search of the ACE2 mutant with the highest affinity to the most common spike protein, we created a library. Following several articles, the mutations were concentrated in 16 amino acids, resulting in over 97,000 variants. The library was reduced by analyzing the predicted score functions in PyRosetta, Swiss model, and MATLAB, giving us only 2,000 variants left for a deeper analysis. For this analysis, we simulated the change in the folding energy of the bound complexes between the variants and the spike protein, searching for the lowest energy compared to the wild-type complex. While working on the PyRosetta model, we simultaneously started working in the lab on expressing the variants in order to check their affinities. For that, we decided to clone a popular variant from our library And we chose yeast because this is a eukaryotic protein, and so bacteria was less suitable. The cloning method we chose is known as any gene, any plasmid, or AGAP for short. It is based on homologous recombination in vivo, by counting on the biological machine that is the yeast cell and overlapping linear fragments. For the purification step, we consulted with some professionals in our faculty, and they advised us to first try regular purification method, relying on nickel column, and if that doesn't work, we should add a dialysis step preceding to the final column step. Unfortunately, we haven't yet got a chance to examine their suggestions. For the affinity test, we'll use two-step fluorescence activated cell sorting. First, we'll take carriers displaying the ACE2 proteins and mix them with spike proteins fused to GFP. The carriers will be sorted to find the ones with the highest fluorescence. The second step of the sorting involves a reaction between those carriers and fluorescent anti-ACE2 antibodies. The complexes will be sorted when the fluorescence intensity correlates to the amount of ACE2 proteins on each carrier. Therefore, the carriers with the highest fluorescence in the first step and the lowest in the second step will be selected as the ones carrying the ACE2 with the highest affinity. And now for the Sybodies. On the quest for an antibody with a high affinity to the spike proteins, we came across Dr. Justin Walter's paper, in which he scanned a large library of synthetic single-domain antibodies against the virus. After consulting with him, we chose to work simultaneously with a couple of Sybodies, designated numbers 15 and 68. For cloning, the Sybody sequences were cloned into a plasmid that was transformed into E. coli KRX. The plasmid enables tight regulation with rhamnose as an inducer. KRX cells are specially engineered to offer high-yield protein purification. 2 days after induction, nickel columns were used for the purification of the proteins. The size of the purified proteins was happily confirmed using SDS page and also Western blot. We talked about the decoy proteins. Now let's talk about the protein carriers.
Cut and action.
A suitable carrier needs to be socially accepted, safe for skin contact, and able to display our decoy proteins on its surface in a strong manner that maintains the protein's viability. Once again, we have two possible directions in which we chose to proceed. From the synthetic biology field, we selected Bacillus-Subtilis spores, and from the biochemical field, beads made from NTA monomers were selected. Let's start with the Bacillus spores. While investigating different approaches, we came across an article about a special display technique known as Bacillus spore surface display, or BSSD, meant for expression of proteins that are known to be foldable in bacteria. Further on, we encountered a study that showed that a functional human ACE2 protein can be expressed and purified from Lactobacillus paracasei. With this in mind, and based on other comprehensive articles, we were able to assemble a protocol for the expression of all our decoy proteins on Bacillus-Subtilis spores. In order to assemble the final plasmid that will be cloned to the bacilli, restriction and Gibson cloning methods were selected, with E. coli top 10 as the chassis. The first step was to insert one of the decoy protein sequences to the plasmid by Gibson assembly. After this, a restriction-based cloning was done to add a linker and a COT protein. COTs are a family of protein expressed during the sporulation in order to create the outer surface of the spore. Once another protein is linked to the COT protein, it will be displayed on the spore surface. The plasmid containing the Sybodies were successfully assembled and verified by sequencing. After the cloning to the bacilli, the decoy proteins fused to the COT proteins were integrated to a specific genomic integration site known as MELOS. Before the cloning process, sporulation was performed as a proof of concept and positive control. Hopefully, once the assembled sequence is integrated, sporulation can be induced and the active decoy proteins will be displayed. If you ever work with bacteria, you probably notice the smell. In a product meant for everyday usage, we wanted to avoid it. Staying on the safe side, we consulted with the well-known pharmaceutical company, Dr. Fisher, a about this issue and others. They have told us that if smell becomes an issue, a bit of perfume should do the trick, as it does in many self-care products. And remember we talked about social acceptance? Well, we realized that the idea of spreading microorganisms on the skin might intimidate some people. Therefore, in our follow-up survey, we included a question confronting this possible taboo. Only to gladly figure it out that most people are okay with it, as long as the microorganisms are harmless. And now for the microgel beads. Firstly, we would like to thank Professor Boaz Mizrahi's lab that advised us and gave us equipment for this part. Originally, the microgel beads, as seen in the microscope images, were created for efficient protein purification. Due to their high-density nickel attribute, they were a great candidate for recombinant histone protein display. The coordinating bond between the nickel and the protein is almost as strong as the covalent bond, so we can rest assured that the decoy protein will remain on the beads. The preparation of the microgel started with fabrication of anti-A monomers. The monomers mixture was evaporated in vacuum, followed by removal of sodium ions. The upcoming step was obtaining the oily anti-A monomers by lyophilization. The obtained monomers were then dissolved in a buffer with other chemicals, creating a watery oily emulsion. Sonication was done to this emulsion, followed by polymerization. Eventually, the polymers were precipitated with methanol, and nickel ions were attached to their surfaces. Eventually, the created beads would be mixed with the purified protein to create the full carrier. For the microgel, while consulting with Dr. Fischer, we raised two potential obstacles. One regarding regulation and the other regarding the product's possible color. The main conclusion of the meeting was that unlike European regulation, the FDA doesn't necessarily forbid the use of bound nickel in an off-the-shelf drug product, such as ours. And for the color, though they doubt that the final product will have any color, if it does, it's not a bug, it's a feature, because you can still cover your entire palm area. And now, for the last piece of the puzzle, for the base of act, we produce thermoresponsive hydrogel. This Puroni gel has the ability to reversibly switch between being completely liquid at low temperature to completely solid at skin temperature. All the components that were mentioned before will be inserted into this hydrogel, creating our product, ACT. Go ahead, Donald.
Thank you, Odas. It might be easier to understand the three-part system that is ACT as a fishing rod, a hook, and a bait. While the SARS-CoV-2 is our great white shark, its baits are the decoy proteins, ACE2 and Sybodies. They are strongly connected to the hooks, which are the Bacillus spores or the microgel beads, and holding it all together to the hands of the fisherman, the hydrogel serves as our fishing rod. To model the effectiveness of ACT, 4 aspects were taken under consideration. First, the diffusion of the viruses onto the hydrogel layer, modeled by using Fick's second law of diffusion. Second, protein kinetics, relying on association constants from the literature. Third, assessed amount of proteins on each carrier. And lastly, the plausible worst-case scenario concentrations of viruses in the air. The partial differential equation was executed in MATLAB. Here you can see the results we came with. The logarithmic x-axis describes the estimated number of carriers on the user's hands, and the logarithmic y-axis describes the initial concentration of viruses in the air. Now, the color gradient describes the relative amount of free viruses left in the air, and the z-axis describes the time required for the reaction to happen. When we have 100,000 carriers on 2 hands, even in the worst case scenario, all the viruses next to the skin would be captured in the gel within 30 seconds. Let's do the math. 50,000 spores per 1 hand means that the carrier's concentration in ACT should be 11,000 spores per milliliter, which is much less than 1 OD. Let's recap. Not that far. Okay. We have 2 options for baits and 2 for hooks. Together, They constitute 4 backup combinations that could go into the hydrogel, creating our product. As for this very moment, we managed to purify and verify the Sybodies and to create both the microgel and the hydrogel. Due to the lockdowns, we didn't get the time to connect them all together and test the product, but they can all assemble ACT. When talking about spores, it is worth mentioning that they can survive for a long time in their dormant state. But when the environment's conditions change, the spores might germinate, making our decoy proteins obsolete. Therefore, we tested the stability of the spores in the hydrogel and in water as a negative control. Here you can see that in 4 degrees, both in the hydrogel and water, most cells were sporulated, and when approaching skin temperatures, the germination becomes significantly visible in both systems. Germination and growth were much more substantial in water rather than hydrogel. No rest for the wicked. We are still facing several issues. The main 2 are: ACT is designed to fight skin transmission, so to achieve good protection, other measures of protection are also needed, like masks and social distancing. Also, to be sure ACT has a long shelf life, we need to make sure the Bacillus spores won't germinate, because vegetative cells don't display our baits. A germination knockout strain is within reach and was used by the iGEM teams from Freiburg in 2016 and Munich in 2012. Since our project focuses on the pandemic, we found it important to raise the juveniles' awareness of the situation in elementary schools. To achieve this goal in a fun and attractive way, we created a Kahoot game. We reached several schools all over the country and made sure they enjoyed the learning process of a stressful subject.
Additionally to that, we kept sharing on social media posts and videos concerning the pandemic, encouraging the people to stay busy and safe at home.
On the 21st of October, we organized an iGEM meetup for several groups, aiming to help each other prepare for the presentations and the QA session. We also invited 3 guests, which are Dr. Omri Drori from TechBio, Dr. Tami Meron from First Arts, and Ilan Breisblatt from the Technion team of iGEM 2019, to lecture about the entrepreneurship world and experience. This year, with all the other Israeli iGEM teams, we set up a video project collaboration meant to aid in the fight against systemic racism in STEM and academia. We focused on 3 underrepresented sectors in Israel: Arabs, ultra-Orthodox Jews, and Ethiopians. A few seconds before we will finish, we wanted to thank all the amazing people and companies that helped us along the way, starting with iGEM, our instructors, and our own team. We are the iGEM 2020 team from the Technion, Israel.
Thank you for watching.
Remember to act.